Hi, my name is Lance Capel, and this is Bio 515, and this is my central nervous system presentation. The topic that I chose is the blood-brain barrier. The blood-brain barrier is a highly selective and specialized interface that separates the circulating blood from the brain's extracellular fluid in the central nervous system. It plays a crucial role in maintaining the brain's microenvironment and protecting it from potentially harmful substances while ensuring a stable and controlled environment for optimal neuronal function. The brain is a privileged site. It's sheltered from the systemic circulation by the blood-brain barrier. It's a highly specialized endothelial structure of the fully differentiated neovascular system. The anatomy of the blood-brain barrier. First, the endothelial cells. Second, the pericytes. Third, capillary basement membrane. And fourth, astrocyte end feet. So, as far as the tight junctions are located between the cerebral endothelial cells. Tight junctions form a highly selective diffusion barrier prohibiting most blood-borne substances from entering the brain. Tight junctions block the pericellular aqueous diffusional pathways between adjacent and endothelial cells. Due to their adhesive function, they seal microvessels and impede the passive diffusion of proteins and polar solutes in and out of the CNS. The astrocyte end feet. The astrocyte end feet ensheath the vessel wall and play a critical role in the induction and maintenance of the tight junction barrier. Astrocyte end feet are not believed to have a barrier function within the mammalian brain. Endothelial cells of the blood-brain barrier differ from those of the rest of the body. Blood-brain barrier endothelial cells have no fenestrations, the more extensive tight junctions, and sparse pinocytic vesicular transport. Endothelial cells' tight junctions limit the pericellular flux of hydrophilic molecules across the blood-brain barrier. Pericytes. Pericytes are embedded within the capillary basement membrane. Pericytes play a crucial role in the angiogenesis, the structural integrity and differentiation of the microvessels, and the formation of the endothelial tight junctions. Their job is to shield the brain from toxic substances, filter harmful compounds from the brain to the bloodstream, and supply brain tissue with nutrients. To do this, the blood-brain barrier has physical tight junctions and metabolic enzyme barriers. Now to the next slide is first, protection. Second, regulation of substance movement. Third, maintaining brain homeostasis. And fourth, protection from hormonal fluctuations. And these are all functions of the blood-brain barrier. So for the protection, it serves as a protective barrier, shielding the brain from harmful substances, pathogens, and toxins that may be present in the bloodstream. Now, this protection helps maintain the brain's delicate balance and prevents damage to neurons and brain tissues. The regulation of substance movement, the blood-brain barrier is selectively permeable, allowing only specific substances essential for brain function, such as oxygen, glucose, and certain nutrients to pass through while restricting the entry of large molecules, pathogens, and most drugs from entering the brain. And the function of maintaining brain homeostasis. The blood-brain barrier regulates the passage of ions, nutrients, and molecules into and out of the brain, maintaining a stable and controlled environment necessary for proper neuronal function and signaling. And last, we go to the protection from hormonal fluctuations. It shields the brain from fluctuations in hormone levels and other physiological changes that occur in the bloodstream, ensuring that these changes do not disrupt brain function. Overall, the blood-brain barrier acts as a crucial defense mechanism for the central nervous system, allowing for proper brain function while protecting it from potentially harmful substances that could negatively impact neurological health. Next here is the transport mechanisms for the blood-brain barrier. If you look at the slide here, I have passive diffusion first, carrier-mediated transport, receptor-mediated transport, active transport, and efflux transporters. Now, the blood-brain barrier employs several transport mechanisms to regulate the passage of substances into and out of the brain's extracellular fluid. These mechanisms include passive diffusion. Now, passive diffusion is small lipophilic molecules such as oxygen, carbon dioxide, and some lipid-soluble drugs. And they can passively diffuse across the blood-brain barrier by moving through lipid-rich cell membranes of the brain's capillary endothelial cells. Now to the carrier-mediated transport. Specific carrier proteins or transporters facilitate the passage of essential nutrients like glucose, amino acids, and certain vitamins from the bloodstream into the brain. For instance, glucose transporters assist in transporting glucose across the blood-brain barrier. Receptor-mediated transport. Some larger molecules such as peptides or proteins required for brain function, for example insulin, can cross the blood-brain barrier through receptor-mediated endocytosis. These molecules bind to specific receptors on the endothelial cells, allowing them to be transported across via vesicles. Active transport. Active transport systems such as the ATP-dependent transporters utilize energy to move molecules against their concentration gradient. These systems enable the transport of certain ions and molecules into or out of the brain, maintaining proper ion balance and preventing the accumulation of harmful substances. And then last, efflux transporters. Now for efflux transporters, these transporters help in removing potentially harmful or unwanted substances from the brain. P-glycoprotein is an example of an efflux transporter that pumps out various drugs and toxins from the brain back into the bloodstream, contributing to drug resistance. These transport mechanisms collectively regulate the passage of substances, allowing essential nutrients to enter the brain while protecting it from harmful or unnecessary compounds. These blood-brain barriers' selective nature ensures a controlled environment necessary for optimal brain function, while maintaining the brain's integrity and protecting it from potentially damaging substances. So these are the properties to cross the blood-brain barrier. Number 1, small size and lipid solubility. 2. Lack of charge or neutral charge. 3. Specific transport mechanisms. 4. Active transport or receptor-mediated transport. And the last is size and shape. So small size and lipid solubility. So substances that can easily dissolve in lipids or fats and are relatively small in size have a higher chance of crossing the blood-brain barrier. Lipid-soluble molecules can diffuse to the lipid-rich membranes of the brain's capillary endothelial cells, thus allowing them to pass more readily. Lack of charge or neutral charge. Charged molecules, especially large ones or those with multiple charges, face difficulty passing through the blood-brain barrier because the barrier is selective against charged substances. Neutral or slightly lipophilic molecules have better chances of crossing. Specific transport mechanisms. Some molecules such as glucose and amino acids require specialized transport systems, like carrier-mediated transports, to cross the blood-brain barrier. These transporters facilitate the movement of essential nutrients into the brain. Active transport or receptor-mediated transport. Certain substances, like some drugs or proteins necessary for brain function, can cross the blood-brain barrier via active transport mechanisms or receptor-mediated transport, utilizing specific receptors or carrier proteins present on the blood-brain barrier endothelial cells. Size and shape. While smaller molecules have a better chance of crossing, certain larger molecules with a specific structure or shape may also have ways to traverse the blood-brain barrier if they can interact with specific transport systems or mechanisms. Overall, the properties that facilitate crossing the blood-brain barrier are related to the substance's size, charge, lipid, solubility, and the presence of specific transport mechanisms or pathways that facilitate their passage into the brain's extracellular fluid while maintaining the integrity and selective nature of the barrier. Molecules that pass through the blood-brain barrier. Oxygen and carbon dioxide, glucose, amino acids, lipid-soluble substances, water, and some drugs. Oxygen and carbon dioxide. Now these are small uncharged gases that can diffuse freely through the lipid-rich membranes of the endothelial cells in the blood-brain barrier. Glucose. Glucose is essential for brain energy. Glucose crosses the blood-brain barrier via specialized glucose transporters that facilitate its transport into the brain. Amino acids. Certain amino acids are crucial for protein synthesis and neurotransmitter production. Enter the brain through carrier-mediated transport systems. Lipid-soluble substances. Small lipophilic molecules or drugs that dissolve in fats can diffuse passively through the lipid bilayer of endothelial cells. While water molecules themselves are small, their passage across the blood-brain barrier also involves specialized water channels called aquaporins. And then for some drugs, certain drugs or medications can be formulated or designed to cross the blood-brain barrier using specific transport mechanisms or carrier-mediated pathways. Overall, the blood-brain barrier plays a crucial role in maintaining the brain's health and functionality by allowing essential substances to enter while protecting it from potentially harmful compounds. Its selective nature ensures a controlled environment necessary for optimal neuronal function within the central nervous system. And just to reiterate, the blood-brain barrier acts as a crucial defense mechanism for the central nervous system, allowing for proper brain function while protecting it from potentially harmful substances that can negatively impact neurological health. The properties that facilitate crossing the blood-brain barrier are related to the substance's size, charge, lipid solubility, and the presence of specific transport mechanisms or pathways that facilitate their passage into the brain's extracellular fluid while maintaining the integrity and selective nature of the barrier. And that was my presentation on the blood-brain barrier. I hope you enjoyed.